Delighted to for us to be here, this lecture is part of our BCG 100 program, which of course marks the centenary of Bacille Calmette-Guérin, or BCG. And if you go onto our website, you can find out about the other activities we're doing to mark this centenary year. So really delighted to welcome our two colleagues today who are going to give us a talk. So Professor Glyn Hewinson is the Sir Cymru Research Chair at the Centre of Excellence for Bovine Tuberculosis for Wales at Aberystwyth University, and he's also lead scientist for bovine tuberculosis at the Animal and Plant Health Agency in the UK. Professor Michele Miller is the South African Chair in Animal Tuberculosis in the Division of Molecular Biology and Human Genetics at Stellenbosch University in South Africa. So welcome to you both. Really delighted to have you both here, and I'm very much looking forward to what you have to say. Thank you very much. I think Glyn is going to kick off.
Yes, thank you, Helen. And I will now try and put my slides up. This is the moment that one gets nervous.
They say we're good to go, Glyn.
Yeah, good. So can you see my can you see the slides, Blakely? Yeah,
absolutely.
Great.
And they're on presentation mode.
Thank you. I can now relax. So I'd certainly like to thank the organisers for inviting me to give this talk to celebrate 100 years of BCG. And also those of you who are dialed in, thank you for giving up your evening to listen to this talk and to join in that celebration of 100 years of use of BCG in humans. I should just say that I hold the Sir Cymru Chair here at Aberystwyth University at the Centre of Excellence for Bovine Tuberculosis. I used to be the Chief Scientist of the Animal and Plant Health Agency at Weybridge, but led their Bovine TB programme for many years. That's no longer the case, but most of the work I am going to talk about is around the work that we did at APHA, and indeed that they are still doing at APHA. And before I move on, I just want to pay tribute to a number of, many people who are involved in this work, and especially Martin Vordermeyer and Gareth Jones, who led the cattle immunology work for developing cattle TB vaccines. Mark Chambers, Des Delahaye, and the team at APHA who worked on the Badger BCG vaccines that I'm going to talk about, and Steve Gordon and colleagues and collaborators at the Institut Pasteur and the Sanger Institute who worked on the molecular biology that you're going to hear about. So I wanted to put that up front just in case we run out of time later, but I'm now going to move on to tell you, to sort of discuss issues around TB and the use of BCG in animals. Does it matter to people, and to give you an idea of what's been going on, certainly in the UK, on the use of BCG in both cattle and badgers. So some of the drivers that we can think about that are going to be happening over the next 40 years, and we're already seeing that, and this slide shows plots from the FAO about what's going to happen in terms of protein consumption, or what they predict will happen with protein consumption around the world over until 2050. And you can see already that there's been huge increases in the consumption of eggs and meat and milk. And we see that continuing past the 2010 prediction there, that by 2050 we will see an increase in about 70% of protein, animal protein requirements. And we're already seeing that in terms of intensification of cattle and dairy farming in developing countries, and indeed places like Ethiopia, which I will mention briefly later, have plans to double their milk output production over 5 years. And of course, with intensification of livestock comes increased risk of zoonotic disease and increased prevalence of diseases like bovine tuberculosis, which, as we know, is a zoonotic disease. If we look at some of the maps of where we see bovine tuberculosis, this is from the OIE reports in 2014 and 15, and it hasn't changed very much since then. One caveat is that this is self-reporting disease, so some countries may have bovine TB that isn't reported. But of the self-reporting these, you can see that bovine tuberculosis is found right across the world. And really, when we think about bovine tuberculosis these days, I would say that we've had lumpers and splitters in terms of the TB complex. Over the past 10 years, we've seen new mycobacterial species being designated in the TB complex, Mycobacterium aereus, Mycobacterium capri, but when I talk about bovine tuberculosis, I'm really talking about any disease that causes TB in humans and as a zoonosis in animals and as a zoonotic infection in humans. And that's one of the reasons that we need to control the disease. If we think about some of the socio-economic impacts of bovine TB, it's not just in terms of transmission of infection from animals to man, but also, as the FAO have pointed out, hunger is one of the most important factors that contributes to the death of people from TB every day worldwide. And some of the estimates are about 5,000 people a day die of TB. That an important factor is hunger in that prognosis. And then if we think about what other things we find that can affect both the economy and the ability to produce protein and hunger, to alleviate hunger, we see that bovine tuberculosis can cause up to 20% reduction in milk production, 30 to 40% of carcass condemnation. In the early 2000s, the agricultural losses were estimated to be at least $3 billion. That will be much more now since the 2000s, where these estimates were made. And of course, as Michele will talk later, bovine tuberculosis, or the Mycobacterium bovis, has got into endangered wildlife species, and of course that has other economic impacts and impacts on health and well-being and tourist industries. Cost of TB control programs are very high in Great Britain at the moment. The costs are about £100 million to government. In Wales, where I am now, it's approximately £24 million per annum just in the control aspects, let alone the losses to industry. And then the WHO have estimated in 2016 that there's about 15,000 cases per year of diseases in humans caused by zoonotic disease, and this leads to about 1,200 deaths annually. And what we're finding also here in Wales and elsewhere is that also the impact of having TB on a farm leads to issues around health and well-being of farmers and their families. And it's one of the greatest causes of stress and mental health issues here in Wales for farmers. In recognizing that we can't end TB unless we end all forms of TB, including zoonotic TB, the FAO, the OIE and the WHO got together a few years ago to consult about creating a roadmap to the elimination of zoonotic tuberculosis. And in 2017, the roadmap for zoonotic tuberculosis was published. There is a link here on the slide. For those who are interested, it's a 10-point plan, of which one was the use of TB vaccines to try and protect cattle and other livestock against infection. And this recognizes that in some countries it's not practical or it's not practical to have expensive test and cull programs, and in other countries it's just not acceptable from religious and moral standpoint. So we now come to celebrate BCG, and I thought this was, given that this is a time of zoonotic disease, we should really celebrate the One Health approach to disease control. And in fact, BCG is a perfect example of this. The Bacille of Calmette-Guérin, or as you'll hear in a minute, it was actually the Bacille Bile of Calmette-Guérin, was actually a One Health approach to TB vaccine development. And here are the two main players: Albert Calmette, who was a physician and was mentored by Louis Pasteur. And the reason I'm telling you about the mentoring is important in the story that I'm about to tell you. And Calmette-Guérin, who was a veterinarian and was mentored by someone called Edmond Nocard at Maison Alfort in the Veterinary College in Paris. And many of you will know, but some of you may not know the story behind the development of BCG. So I thought I would spend a couple of minutes just sort of recounting the story of how BCG was developed. So in 1903, Emil von Behring, a famous a Nobel Prize-winning scientist, postulated that pulmonary tuberculosis in adults could result from intestinal infection during childhood. And that was partly because a lot of childhood TB, it was thought, was caused by drinking infected milk infected with Mycobacterium bovis. And in order to test this hypothesis, Calmette-Guérin set about investigating that hypothesis. And they used M. bovis because, one, it was likely to be an intestinal pathogen, and two, it was likely that infection might be acquired through milk. And in 1905, they showed that fine emulsions of Mycobacterium bovis delivered into the rumen of cattle or goats showed disease in the lungs, so that they proved that hypothesis that if you gave M. bovis into the rumen, then you could develop diseases, tuberculosis disease in the lung later, due to the sort of spread of disease from the gut to the lungs. But in order to do so, the suspensions had to be very fine. And so the way that they made sure that their suspensions of M. bovis were fine was that they used beef bile as a detergent to break up the clumps of M. bovis. And when they did this, they also noticed that the media attenuated the M. bovis. And this was the Bacille Bile, or the bile bacillus. And they noted that it was no longer as virulent in animals as it had been when they first isolated it. Now, the M. bovis that they used was derived from Nocard, and as we saw, Guérin was a student of Nocard at Maison Alfort. And because Pasteur had shown that you could attenuate different bacteria by growing them on culture, then they combined the observations that you could attenuate M. bovis using ox bile with the idea of repeated subculture, which Calmette had obviously been exposed to through Louis Pasteur. And they then subcultured M. bovis in media that contained beef bile and glycerol over a 13-year period. And the other thing I should say about One Health approach is that before they started using BCG in cattle, in humans, they used it in cattle. And this picture can be is a model that was produced celebrating the first BCG vaccination experiments that Calmette-Guérin did in cattle in the Institut Pasteur in Lille in 1912. And here you have a schematic model of that first trial that celebrates the success. So all the cows that are standing up are the cows that had received BCG vaccination, a fairly high dose of BCG vaccination, but then the animals were challenged with a very high dose intravenously of M. bovis, so that 30 to 40 days afterwards, you can see that the cow right on the far left is part of the control group that didn't receive BCG. And if you turn the model the other way around, so it's a three-dimensional model, all the cows on the other side, which are the control cows, are lying down, obviously sick and dying. So 30 to 40 days after intravenous injection with M. bovis, the cattle that hadn't been vaccinated with BCG had disseminated disease, were clinically sick, whereas those that had received BCG were happy, standing and feeding. And although at post-mortem they could still find M. bovis within some of the tissues, there was much reduced lesions, and animals were not sick as the control animals were. And that was the first vaccine experiments in 1912, so slightly over 100 years since the use of BCG in cattle. And so as a result of this, actually BCG became quite used widespreadly across Europe and other countries. From the 1920s really to the 1960s. And the interesting thing is, if we do some meta-analysis of the variability of the efficacy of BCG used in the field, we find that just as in humans, we see in some instances it's highly effective, and in some instances BCG really is not protective at all. We see the same things being mirrored in some of the cattle trials that were reported. So you see anything from a 70% protection all the way down to no protection, for instance, in Malawi. Now, it's very difficult to work out exactly what the reasons for this variation is. It could be that there was different exposures to environmental mycobacteria. It could be that cattle were already infected with TB before they were vaccinated. It could be to do with force of infection. It could be to do with the strain of BCG used. And it could be to do with the readouts that were used to give that vaccine efficacy. But whatever those reasons, it was clear that BCG use in cattle gave variable efficacies, and that there was very little standardization of BCG in cattle. Some of the results from the UK in the 1940s and 50s suggested about 50% protection, and indeed a number of studies showed that you could eradicate TB from a cattle herd, but it was quite an inefficient using BCG vaccination, but it took about 7 or 8 years to do so. And so when Bang in Scandinavia did his experiments to show that it was far quicker and more effective way. As I was saying, the cattle trials were variable, and once it was shown that test and cull ways of controlling TB were more efficient than BCG vaccination, we stopped the use of BCG because BCG sensitized to the tuberculin skin test, so we couldn't tell which animals were protected and which animals were infected. And then in joining the EU, that was put into law that it was then illegal for the UK to vaccinate against bovine tuberculosis. However, obviously vaccination did show some promise. And so for the past 15 to 20 years, we've been looking at how we might improve BCG vaccination to help with TB control, not just in the UK, but as I was saying, around the world. There are obviously many countries that can't afford test and cull strategies for TB control. What I would say is, to date, the most promising approaches require BCG. We haven't found any vaccines that are better than BCG. But along the way, we managed to optimize the use of BCG in cattle and found that if you use BCG in this optimized form, or use it in a heterologous BCG prime boost strategy, for instance, by combining with recombinant adenoviruses, you could get some improvement on BCG. But of course, both BCG or BCG prime boost strategies sensitize cattle to the tuberculin skin test, and therefore, unless you can develop a diagnostic test that can work in the face of BCG vaccination, then it's not possible to use this TB vaccination, or at least to use BCG in countries that do the test and slaughter strategies for TB control. In terms of optimization of BCG vaccination, where we have got to in the UK is that we use the strain BCG Danish 1331. We use 5 times the human dose, and we use it subcutaneously. And when you do that, you find that experimentally, when you challenge animals with TB via the M. bovis, via the intratracheal route, you get about a 70% reduction in visible pathology scores, and that duration of immunity lasts between 1 and 2 years, which was actually the sort of observations they found in the 40s and 50s in terms of when immunity waned in cattle. Of that 70% reduction, we see a full spectrum of protection. We see full protection from disease in some animals. We see partial protection, which is reduced pathology scores compared to vaccinates, and in about 10% of animals, but it does vary from experiment to experiment, we see pathology scores that are similar to non-vaccinated controls. When you go out into the field, because of course with experimental challenge, this is a very harsh test of a vaccine, which doesn't necessarily always predict what you'll see in the field. Using some of these optimization of dose and route, we've now, over the past few years, there's been a number of natural transmission studies across the world, and I've just listed a few here. So there's been a couple of experiments where vaccinated and non-vaccinated animals were introduced into 100% reactor herds in Ethiopia, and they found that after 10 to 18 months, there was a 61% efficacy against efficacy in which there was no, in that 60% of animals, there was no observed pathology in the vaccinated animals. It also suggested that the duration of immunity might be longer in the field than the minimum 1 year that was demonstrated experimentally, and we think this may be because you are constantly exposed to infection, therefore topping up your immunity in the field. A second repeat experiment showed a 30% efficacy. So here we see variation again. And here the severity of disease was much heavier in this herd. So it may be that the force of infection was much higher. And if you think about it, you're asking a vaccine to protect an individual animal day in, day out, whatever their immune status, whatever the dose that they're exposed to. And it may be that force of infection does play a role in vaccine efficacy. The severity of pathology was, as I say, the severity of pathology and dissemination of M. bovis was lower in the vaccinated infected animals compared to the non-vaccinated animals. And so a third experiment now is ongoing to look at the effect of BCG vaccination on onward transmission. So would those vaccinated animals that acquired M. bovis go on to then infect non-infected individuals, which is of course important if you're thinking about TB control through vaccination. Other encouraging results have come from New Zealand, where they've seen 80% efficacy subcutaneously and about 67% efficacy through oral vaccination. In Mexico, the use of BCG Tokyo subcutaneously gave 59% efficacy, And in Chile, in a recent publication in which Martin Vordermeyer was a collaborator, BCG Russia gave us 66% efficacy. And also, interestingly, there was evidence of beneficial effects on milk yield in the animals that received the BCG vaccination. However, as I said, if we're to use BCG in countries that require test and slaughter strategies to run alongside vaccination, because we recognize that vaccines won't be perfect, you need these so-called DIVA tests, which detect infected animals amongst vaccinated ones. And there are a number of approaches that we have been doing and one can use to try and develop such tests. You can modify existing diagnostic test platforms, such as gamma interferon release assays, or indeed the tuberculin skin test. And the strategy that we've used is to use defined antigens, which target the immune system of M. bovis infected animals, but not in BCG vaccinated animals. And I'll go into how we do that in the next couple of slides. And what we found here is that the skin test format for these defined antigens promise a higher specificity than the gamma interferon based assays. So we were very lucky when we started this work that the genomes were either being sequenced or had been completed, and of course the M. bovis genome and the BCG genome were sequenced in collaboration between APHA and the Sanger Institute and the Institut Pasteur. And so we could start comparing genomes of these different species of mycobacteria to try and identify antigens that might be missing from BCG that we could use for our diagnostic tests. And as many of you will know, when Calmette and Guérin were doing those passages of the M. bovis on the bile and glycerol media, a number of things happened to BCG. It became adapted to a life on glycerol, and it also lost a number of areas of its genome called these regions of difference. And within those regions of difference, two highly immunogenic proteins exist, which are CFP10 and ESAT6, and these turn out to be very good diagnostic antigens. What we did was that we mined all the antigens that were missing from BCG, that had been deleted from BCG, using infected cows, and we found that we got different responses to different antigens, and this is just an example of the sorts of screening processes that we did using gamma interferon assays as a readout for immunogenicity of these different genes that were missing from BCG. And we found that the ESAT6 family and the secreted antigens were highly immunogenic. And after much work, I'm condensing 15, 20 years of work into a couple of slides, we found that a number of defined antigens targeted the immune systems of M. bovis infected animals, but not those that were animals that were vaccinated with BCG. And the three antigens that gave the most promise when used together were ESAT6, CFP10, and this gene called RV3615C, which is present in BCG, but because it's lost the secretion mechanism, it's not secreted and therefore is not recognized during infection. This is a very busy slide, but these are the sort of data so far that have been used to evaluate skin test, the defined skin test, the DST, to be used in conjunction with BCG as a DIVA test. And what you can see from the first two rows are specificity, and the second two rows are sensitivity of these reagents compared with the skin tests that are used using tuberculin. And in the UK, we use this comparative skin test. And you can see that the specificity of the defined skin test using these antigens is 100% so far in over 200 BCG vaccinated animals and 136 unvaccinated controls. And when we look at sensitivity of these antigens, we find that this defined skin test is actually more sensitive than the comparative skin test in experimentally or naturally infected animals. And at the moment, the confidence intervals overlap between BCG vaccinated and in bovis infected animals. And so more work is being done to see, to look at the true sensitivity of the skin test in animals that have been vaccinated but go on to get infection with M. bovis. And I should say that the target specificity that has been set for this skin test to be used in the UK is 99.85%, which is based on cost-benefit analysis. So at the moment, given this data, the defined skin test is looking very promising in the preliminary results in the UK. But also when this has been tried out in Ethiopia, and here the DSTF or the DSTC that you see here are the 3 antigens used either as a fusion protein or a cocktail of proteins. And we find that the specificity is very good in Ethiopia compared to the comparative skin test. And this is really because there's more environmental mycobacteria that mask the skin test response to bovine tuberculin. And also that the sensitivity is higher of the defined skin test than the comparative skin test, again, because of the strong responses to the avian tuberculin. and so I think the sensitivity of the comparative test here is about 70% compared to 90% in using the defined skin test. So all that looks very promising. What happens though in the field? And as many of you will know, we've now there were now announcements made by DEFRA that there will now be some field trials of both the defined skin test and cattle BCG, which are due to start shortly. There are a number of phases with stop-go decision points. The first is to see whether the defined skin test is as specific in the field as it is so far in the results that I showed you, and whether it's safe because we are injecting a cocktail of or a fusion protein into the animal. The second phase is a larger safety trial for BCG and for in combination with the skin test, and that will confirm the target specificity in the vaccinated BCG vaccinated animals. Once that's completed, that will give data that allows application for a marketing authorization. And then if that's successful, the cattle BCG and defined skin test will be rolled out. And the time scales that are envisaged there, if all goes well, is about 5 years. I just want to quickly touch on wildlife vaccination. I have only 3 slides on this because I know Michele will talk much more about wildlife vaccination. One of the problems for countries that haven't been able to eradicate TB is that once you get bovine tuberculosis into a wildlife reservoir, it's very much harder to eradicate the disease. And so once you have TB in a wildlife reservoir, what do? Well, there are 3 options. You remove the source of infected animals, and you can see in England at the moment that's being done by the controversial badger culls. You can prevent cattle coming into contact with infected wildlife or environment contaminant, the environment that's contaminated with M. bovis from infected wildlife, so-called biosecurity measures, but those are often challenging and difficult to know what measures are most effective. Or you can vaccinate cattle and or wildlife with a vaccine. And as I said before, BCG is the most promising vaccine so far. So capturing over 10 years work in one slide, apologies to those who are involved with this. Badger Bcg has been granted a limited marketing authorization for use intramuscularly in badgers. It was evaluated both experimentally and in the field during a 4-year field study in Gloucestershire. And although vaccination with BCG doesn't give you perfect protection, as I said, no vaccine does, vaccination was shown experimentally to reduce the severity and numbers of lesions and to reduce excretion of M. bovis, which is really what you want to do, is to reduce the environmental load and onward transmission of infection. And so the three main findings is experimentally vaccination reduces the likelihood of badgers developing lesions or excreting TB. In the field, vaccination reduced the rate of new infections, which were measured using diagnostic tests, by 76% in the trials in Gloucestershire, and these were done by vaccinating social groups of badgers and comparing the TB found in those badgers compared to social groups that weren't vaccinated. And also in those studies, they found that vaccinating more than a third of animals in a badger social group reduces the new infections measured by the diagnostic tests in unvaccinated badger cubs by 79%. So you were seeing that you had reduced onward transmission to cubs before they had emerged from under the ground. And so I think this is very promising evidence for effect of BCG reducing TB in badgers. And this was also mirrored in trials in Republic of Ireland in terms of looking at the effect of BCG vaccination in badgers in the field. The one thing that I think certainly the farming communities want to know is, it's all very well reducing infection in badgers, what happens to infection in cattle? And there's been a publication recently from the Republic of Ireland which looked at a non-inferiority trial in Ireland, which ran between 2011 and 2017, which was designed to evaluate if vaccination of badgers was as good as continuing with badger culling. And the results here were found that vaccination was as effective as long-term continuous culling in lowering cattle TB incidence in 4 of the counties that they looked at, ambivalent in 1 and not as effective in 2 counties. And the proviso here is that culling had already happened, so it looks as though vaccinating badgers might well be a good exit strategy from culling, but what we still don't know is whether you could use vaccination of badgers to replace culling before culling happens. And that data, I think, really does need to be looked at. And 2 large, not trials, but 2 areas of the country are now are either about to or are undergoing badger vaccination in the Gower Coast in Wales and are about to start in Sussex, which was announced recently. So that's really all from me. I just want to pay tribute to huge amounts of international collaboration that has got us to this point and given us more insights into how BCG may or may not be used to control TB in cattle and livestock and badgers, and to thank all the key collaborators and funders for all the wonderful work and fun in doing this work that we've shared through the years. Thank you very much. I'll hand over to Michele now.
Thank you, Glyn. I'm just going to turn my camera on quickly for a moment to say hello to everyone that I may not have met previously, and welcome from myself. I'm based in the Kruger National Park in South Africa, and a shout out to all of my wonderful young research group that's based at Stellenbosch University, and the many colleagues I see on the call. So I'm looking forward to having a discussion with everyone. So let me just share my screen. Oops, hang on. Okay, confirm you can see my slides?
Yep, perfect. Thanks, Michele.
Thank you. So again, I'm just going to continue on the theme that Glyn has started and done an incredible job describing. He's, I think, covered most of the points I was going to make, but in contrast, what I wanted to do was really try and evoke some thoughts about BCG as a vaccine in animals, and kind of the pros and cons and what we have to look forward to in the future. So I was very honored to be asked to speak in the same event with Glyn. He's one of the rock stars of Bovine TB, and I also see many of the colleagues in the audience as well. So thank you for spending some time with us. So again, I think the history is very important in where we've come from and looking at where we're going forward. And Glyn has described that importance. And I think oftentimes we forget that although human TB is a global burden in the current days, bovine TB was also a very significant cause of death around the world before the times of TB control programs in animals and pasteurization and those type of things. So TB was responsible for about 1 in 4 deaths in the 19th century. So you can see that physicians and veterinarians were the appropriate people to really take on this challenge of trying to find ways to prevent that. And of course, we've heard about the development of BCG and keeping in mind that it is a live attenuated vaccine. There's some pros and cons to think about that, but I think again it really highlights how people with expertise from different fields can really make significant advances in science and medicine and other fields. So as Glyn mentioned, they really believed that human infants were possibly being infected orally. And so it was 1921 when the first oral dose of BCG was given to an infant whose mother had just died of TB. And that really started the advent of BCG vaccination programs around the world. Of course, now it is primarily administered as an intradermal injection in healthy infants. And in this slide, you can see with the blue colors and the orange being historically vaccinated, the majority of the world still continues to receive BCG vaccines to protect human infants. And it is very effective in reducing infant mortality, primarily from TB meningitis. So because of that, the WHO recommends that newborns in countries with high TB burdens do get vaccinated as part of their program. The interesting thing about BCG vaccines is they're not only used for protecting against TB, but it has been used for other treatments, including instillation into the bladder for bladder cancer in humans. And there have been some studies with the suggestion that BCG vaccination can train the immune system to actually protect against other respiratory viruses and possibly even COVID. So I know a lot of those studies are ongoing, and that is somewhat up for debate. But as Glyn mentioned, no vaccine is perfect. And we have to think about when we're trying to decide in a vaccination program, what are the pros and cons of a particular product, a route, the target population, and those type of things. So as you can see, BCG efficacy is variable. And I'm talking primarily about humans, but also we've seen that in the cattle studies and on other animal models. And there's a lot of different factors that might explain that. One is that different strains of BCG are used in vaccines around the world. In different areas, age at vaccination, exposure to environmental mycobacteria, the route of administration, and in some studies they even say that there's a difference based on the latitude of the country which is using that vaccine. So there's a lot of work that still needs to be done to try and understand how the host's response can be modified so that BCG has a better efficacy. Because it is a live vaccine, it is a potential risk if you administer it to immunosuppressed people. So at this point, recommendations are that people that are immunosuppressed not receive the vaccine. And as we saw earlier in one of the meta-analyses, it showed that BCG generally, if you put all of these studies together, gave about 50% protection from different forms of TB across different age groups in people. So again, there's a lot of things that need to be explored, and I know it's, a lot of work is going into that. But we know that TB is a complex disease in people. We still don't understand how one individual might become infected and clears the infection, whereas another individual becomes latently infected or someone else progresses to disease. So there's a lot about this disease that's very complex in a single species. And then when we start to be talking about TB in animals and multiple species, it becomes an even more complex problem to solve. So Glyn showed you a slide of this already, just emphasizing the fact that TB in animals is a global issue. I know people in the US, for instance, often don't really think of it as a major concern, since there are pockets of infected populations, both in people and in animals. But in places like the UK, certainly in Africa and Asia and South America, it is still a major global concern. And this is both for domestic animals and in livestock. So again, the reason that why should we care about animal TB? We don't care about, parvovirus in dogs or, dermatitis in your cat or things like that, unless you're the owner of that animal. But really, animal TB is a disease that affects at so many different levels when we're talking about the impact on us and our community. So, of course, we know that it has the impact on animals. They can get sick, they can die. It can be transmitted to people, and that's really was the impetus historically for controlling it in animals, in cattle in particular, was to protect humans. It really wasn't about protecting the animals. But now we know a little bit more about this, and the fact is it's not just about a disease that makes animals and people sick and sometimes die. It really has a lot more widespread ramifications. So if your cow gets TB, oftentimes they have chronic weight loss. They lose productivity. In parts of the world where communities use livestock as kind of their savings account, like in Africa, people put money into buying cattle instead of putting it into a savings account. If your cattle gets sick and then spread that to your other cattle, that's potentially your life savings and your family's income severely affected. Of course, that also means that results in loss of food security through decreased production. It threatens the livelihoods of not only the livestock owners, but on a larger scale, when countries are deemed to have particular diseases such as bovine TB, it changes their trade status in terms of the value of their agricultural products. And so again, it not only affects the individual farmer, it affects the economy of a nation. So again, it has so many different complications that it's no wonder that we should really care about this. And of course, when we're talking about valuable livestock, breeding stock, or endangered species, when you have animals that get sick and die or have to be euthanized because they are infected, you can lose genetic material that, especially in small populations, you may not be able to recover. So again, Glyn mentioned this. I think this was really a key turning point in many ways for looking at bovine TB, not just as an animal problem, but as a human problem, is when the roadmap for zoonotic tuberculosis was published in 2017. It had this plan, and I mean, I think it looks great on paper, but you know, you have to start at the real basics in many cases, and that, of course, from the standpoint of many of us, the first thing we need to do is get the disease under control in animals, because as Glyn mentioned, if you have a reservoir like the badgers or buffalo in South Africa or white-tailed deer in Michigan in the United States, you'll never be able to do any of these other things and get this program to be a success. So there are lots of control programs for animal TB in many countries around the world. And again, the reason that many of those were put in place was primarily to protect people and to protect agriculture. And these strategies really were about preventing spread from animals to people and also spread between animals, particularly, you know, between herds of cattle in a country, for instance. But what we often forget is that these diseases, bovine TB, of course, we know can go to people and cause zoonotic TB, but human TB can also be transmitted to animals. And that can have, again, an impact in terms of, you know, the same effects that we talked about with the cattle. If you have a cattle herd in a community in South Africa that gets infected with MTB, the consequences are going to be the same. It doesn't matter to them whether it was bovine or human. And even now we're seeing this with some of our endangered species, such as human tuberculosis being found in Asian elephants, both working elephants. It's been discovered in free-ranging elephants. And we've also found for the first time M. tuberculosis as the cause of death in a free-ranging bull elephant in Kruger National Park. So the impacts of human actions on animal disease is also something that really needs more awareness. One of the problems is these programs, of course, are very complex. They're very costly. They take a lot of people, a lot of experts in a lot of different fields. And Glyn mentioned all the years that they've been working in the UK with, you know, top scientists, epidemiologists, veterinarians, human public health officials, agencies, etc. And it's still an issue. And one of the things that, you know, what it boils down to in many of these programs is that if you find an infected animal, it is culled, it is euthanized. Generally, the TB programs for animals, unlike for humans, do not allow treatment of those animals. So again, it's not only loss of the productivity and the value of that animal, it's a loss of life for that particular individual. So it's really quite a serious step if you find an animal that's infected. So with all that said, and what Glyn has shown, gosh, why aren't we using this BCG vaccine in animals? You know, even if it's, let's say, 50% effective or 25% effective, or it can reduce the, you know, number of infected herds over time, gosh, we should be using it. And we know vaccines have improved the health and saved human and animal suffering as well as lives. So I think COVID has really shown us how important science is in creating these tools for us to control disease. But I think the other part that we don't often think about is we need to understand what is the goal of vaccination and what are the pros and cons that go with that decision when it comes down to whether it's a public health program or one for animal health. So I'm going to go through a few of these things of why we haven't been using BCG, just to provoke some thought and discussion. And as Glyn said, one of the big things is we are using immunological tests to identify infected animals. So that means that we do a skin test, or in the case of primates, we inject some of the proteins from the bacilli under the skin, and we're looking for an immune reaction, basically, swelling, heat, redness, signs of inflammation. And what that is it's stimulating a memory response in that animal. So in fact, that is how vaccines work, is you're basically stimulating the immune system so that the next time that animal sees that particular pathogen, that they're going to have a much more robust immune response. So if you give a vaccine or you are infected, using specific types of tests that we have for animals doesn't allow us to distinguish between the vaccinated and the infected animals. And that's true whether it's a blood test or a skin test. Now, as Glyn said, they have made tremendous progress in getting around that. And I think that's really key. And obviously we'll all be watching very closely over the next few years how those trials go. So this may or may not be a major issue. For the most part, what we know about BCG vaccination is that it's done in young animals, it's done in infants or children to get that effective response. Well, that's wonderful because when a human is born, you have a birth date, but when you have animals, on a farm, you can identify and say, okay, these animals are all 1 month of age or those type of things. But if you're dealing with wild animals, it's very difficult to oftentimes even know that they've been born, to age them, to access them. And if we think that we have to give this vaccine within the first, few months of life, that makes it much more logistically challenging for us to think about vaccinating animals as a group, as a population. And of course, animals age differently. So what might be fine for a calf up to 1 year of age, for a mongoose or a badger, maybe it's only the first 2 months of life where that vaccine is really going to be most effective. So we have a lot of knowledge gaps around this. The other thing is, every year we're finding more and more species that are reported to have tuberculosis or infection with one of the M. tuberculosis complex organisms. And this continues to grow as our ability to detect infection improves. So, we have elephants, we have rhinos, we have fur seals, we have wild dogs, we have possums. And if we're going to do a one vaccine fits all approach, that's going to be really difficult because we know that the immune responses in these species can vary significantly. And so we really have to understand how each of these taxonomic groups are going to respond to the BCG, what is going to be an effective or a protective immune response, and whether they even will develop an immune response to the different components of the vaccine. This was a great little diagram I found recently in a publication, and I just thought it visually represented kind of the complexity of when we're talking about TB in mammals. And so you can see we have humans and cows down here in the kind of the left bottom of the diagram. And so much of our literature is focused on humans and cows and laboratory species as models. But in fact, TB is not caused by one organism. It's caused by a complex of organisms that are closely genetically related. And of course, as we talked about, MTB and M. bovis are the most common ones we find. But then there's some really unique members of this group of organisms that only infect a specific species. So Mycobacterium cercarii only infects, that we know of, meerkats. So what is it different about that bacteria that it only causes disease in one host? And what does that mean about the immune response to that particular species to other members of the complex? So lots and lots of questions. And as you can see, it's quite a complex diagram to think about. Well, how do I use BCG to protect a gorilla against, M. africanum, or how do I protect a cow from M. bovis? So I think, again, as our techniques develop and we learn more, we also have more questions. As I mentioned, we have all these different species, and something that we do when we want to use a vaccine is you want to find out, is it safe, first of all, and then secondly, is it effective? And when it comes to that, again, we're looking at the immune response of that particular species, which could vary significantly. And not only that, you need to think about the age, the route, the dose, the strain, what tests are available to find out if that giraffe that was vaccinated is protected. So it's really quite a complex thing. The other thing, and I know Glyn brought this up, and I think it's really important, oftentimes we forget, is that vaccine efficacy can be measured in different ways. So again, we got to go back to thinking about what is the goal of the vaccine program. Do we only want to protect against disease, or do we want to protect against infection? And those can be different things. I think that oftentimes when you protect against disease, you are also cutting down on the shedding, the contamination that comes with disease. But in fact, if an animal that has fewer lesions but is still infected is shedding, then that vaccine isn't really achieving the purpose of stopping transmission. So unlike in humans, where we want to protect the health of the individual, with animals, oftentimes it's a population based decision. And we need to think about how that differs in terms of the goals of the vaccination program. So even if we get all of that to work, I think, once again, COVID has really shown the world the cost and logistical challenges associated with rolling out a vaccine program. So, human life obviously doesn't have a price. But when it comes to animals, especially production animals, farmers often have to make a decision. Do I spend $1 on that vaccine, or do I just cull that animal if it does, get infected or get sick? And so there's some really hard economic decisions, as well as moral and welfare issues that have to be considered when you're going to vaccinate an animal. We talked about, in some cases, oral vaccination may be more suitable for vaccinating a particular species. So then you have to think about, how do we formulate this? How do we make it appealing so that the target species consumes it, but other accidental species don't, because you may not know the safety in those other species. When it comes to individual animal vaccination, cattle have ear tags and microchips and So picking up where we left off, one of the things that we know about vaccination programs is that they're quite expensive, requires a lot of organizational resources and human capacity in order to implement them. So oftentimes in areas where we see a lot of both animal TB and human TB, these are countries that are low and middle income countries. So for example, with this map in the left-hand corner, you can see that although people are aware of animal TB control programs around the world, having those doesn't necessarily mean that they're actually implemented, and that is typically done because of lack of finances, funds, and capacity. So in Africa, there's actually very few countries that apply these programs in a routine manner. There's a lot of reasons for that, but primarily one of those is, again, lack of funding. So in the right-hand corner, you can see a graph. The animal TB program from some sources runs about close to $40 million a year in the US. And this is a country with very few cases of animal TB. In the UK, as we heard from Glyn, they have put a lot of resources into this. And one of the sources I saw, it was about 66 million US dollars as comparison, again, because they've really taken this seriously and trying to address the control of that. And in contrast, in South Africa, the entire animal disease control program budget is only $3 million. And this is in a country that is 5 times the size of the UK and has numerous controlled animal diseases that they are trying to address, including things like African swine fever, brucellosis, foot and mouth disease, rabies, etc. So it's not only resources for bovine TB, it's also for these other diseases. And you can see that sometimes that money isn't going to go very far. The other thing, again, that we need to keep in mind is this is a modified live virus. And so we have to be concerned about safety issues for accidental exposure of humans and also other non-target species. So the vaccine can persist in tissues and secretions for a period of time. And so the humans that are handling that animal may be exposed to the live organisms. In most cases, this isn't going to cause a problem, or it might result in localized infection. But if you have people that are immunocompromised for any reason, which we often see in low and middle income countries due to malnutrition, the co-infection with HIV/AIDS or other conditions, this could potentially lead to a more serious infection. And again, it comes back to the ability to identify animals that have been vaccinated to be able to put in place safety precautions and making people aware of that potential exposure. Again, in order to really roll out a BCG vaccination program, there will have to be buy-in from a country's health agencies, animal health agencies. And so it's really up to each of those countries to assess the goals of that vaccination program and regulate how BCG is administered and to what animals. Again, there have been some positive examples with baits containing oral BCG that have been implemented in vaccinating possums in New Zealand. Another example of a wildlife reservoir, bovine TB, badgers in Ireland, as we heard, and also wild boar in Spain. So at this time, unfortunately, the World Organization for Animal Health, the OIE, currently generally advises against vaccination of cattle against bovine TB. However, as we heard from Glyn, there are some studies that are going to be rolling out in the next few years, looking at both the safety and efficacy of BCG vaccine in cattle and the ability of those DIVA tests to distinguish between vaccinated and infected animals under field conditions. So that's a very exciting development. It's going to be important that a lot of research is done concurrently in developing these assays for use by regulatory animal health agencies. And of course, that's going to require an investment in research and implementation of these programs. So once again, as we can see from that previous slide with the cost of TB programs, it may not be economically feasible in some of the more in the developing versus developed countries. But I want to finish up with some good news. There's been a lot of work by many people, including some of our colleagues that are on this call, in looking at how to use BCG in vaccinating many different animal species and how it can be administered, its efficacy, its safety, and ability to help protect against TB in these species. So I think that it's a very exciting time for this field, and really looking forward to the positive outcomes of some of these studies that are ongoing. So in summary, I do think that BCG vaccination or another type of vaccine can play a role in a comprehensive animal TB program. However, for this to really be effective and importantly cost effective, it may require selection of certain populations that can be monitored and therefore benefit from vaccination. And that would probably be, in many cases, livestock in the beginning. Of course, we need to get regulatory approval before any of this can be rolled out. So that has to be happening simultaneously with the implementation, and of course, the research to monitor and be sure that we're achieving our vaccination program goals. This is a great opportunity for research, including basic research on comparative immune responses in different species, improving our ability to detect TB, again in different species, and looking at transmission, especially at interfaces, and of course getting quite creative with vaccine delivery, safety, and efficacy in wildlife.
So we've got a couple of questions in the chat, and I can see that Colin has his hand up, but actually I'm going to use Chair's prerogative and start, which is, so it's really interesting and delighted to hear about the defined skin test and, you know, hopefully moving forward to using BCG in cattle. Presumably that could be eventually globally as well as just in the UK. I'm thinking about Michelle's, where she left us, thinking about vaccinating other species of animals with BCG. My question is really about the economy or the economics of that, and also the manufacturing capability, because I know that you said BCG dose in cattle is higher than in humans, and there have, in the last 20 years I can think of at least 3 significant periods of time where the world didn't have enough BCG to vaccinate humans. Can we really make enough BCG to vaccinate animals, and how is that ever going to be cost effective?
So I think that's a good challenge, Helen, because even the badger BCG vaccination program in Wales was halted when we had a shortage of BCG for infants. So I think that is going to be a really big challenge. One thing I didn't say was that I agree with Michele, you have to optimize the dose for the different species. So for badgers, it's 10 times the human dose. For cattle, it was 5 times. So again, that becomes really in terms of cost. So I know that a number of BCG manufacturers were bitten by the problems they had in producing human childhood BCG. And I know that they are, or the last time I spoke to them, I know they're trying to up their game. But I think it's still a challenge. I mean, although it's cheap, it has to be even cheaper for animals than humans.
Exactly.
And certainly the test and cull is just not possible in developing countries. So you need a very cheap vaccine, and you need the logistics to disseminate that vaccine as well.
Yes, and I guess it then comes down to the efficacy, doesn't it? Because unless your vaccine is very effective, you can't abandon other methods of control.
No, you have to. You have to get coverage, and you have to have a good efficacy. And that I think a lot. And I saw that Mart had put in the chat. We really need to know. So for animals, it's a population-based thing. So the question is not just, does it protect animals that are in contact with infected individuals, but what is the onward transmission of those that have been exposed? And experiments are cooking, but we won't know for a while.
Yes. And I can see Joe has put in the chat, high titers and standardization could be achieved by fermentation. It's absolutely the case that I think up until recently, there's been no investment in manufacturing process for BCG for any species. And clearly a bit more of that is needed. Now, I'm just going to try and be democratic and do this in order. So
pick up on that fermentation. I know there were discussions with manufacturers about fermentation, but it becomes a different product, and therefore you have to go through the whole licensing safety again, because I think there was some studies where they swapped a pellicle grown BCG for a liquid grown BCG, and you had more reactogenicity was observed in Scandinavia, I think.
Yes, no, that's absolutely right. Lots of human data on that. Okay, so I've got a few questions. Let's rattle through them. So Susie, first of all, has put in the chat, if BCG is used to vaccinate cattle, will it not now be a challenge to distinguish BCG vaccinated cattle from tuberculosis infected cattle, particularly since infected cattle have to be culled? So I think that's your point about the defined skin test, isn't it?
Yeah, so you need, yes, if you're going to vaccinate, you need a defined skin test, or you don't do your test and cull. Although some of the data suggests that the sensitization wanes at about 9 to 12 months.
Yes.
And the duration of immunity is a year, so you have to boost
yes,
after a year. So that the timing of testing and vaccination would also be a challenge.
Not trivial, is it? We think we have it hard in humans. It strikes me cattle are even harder. Okay, so we've got a question from Sanjay Kajira, who's got his hand up.
You should be able to, Sanjay, you should be able to turn on your mic and camera.
Hi. First of all, in all transparency, I work for AJ Vaccines. We manufacture BCG vaccine. So first of all, transparency from that perspective. This was a very, very interesting session and quite an eye opener for us. We do manufacture BCG vaccine, which is WHO pre-qualified, and it is used in UK. And I was very surprised when I joined this company, and UK was the only country where we are actually we have license to sort of sell it from an animal perspective. What are your thoughts? I have a couple of questions. Number one, what are your thoughts in terms of a WHO pre-qualified vaccine being used across board in different countries? Do you think that is something that we should raise at some point in time at different levels from an authorization perspective? Number two, the second question is, there is a lot of discussion when it comes to human BCG vaccination when there are different strains, and there is a lot of debate from a perspective that which strain is better than the other. I assume that is not the case in cattle. And thirdly, thank you very much for this. I really enjoyed the session, and post the session, if I would be happy to connect with some of you to take this forward and see how we can help from our platform in any way to support cattle BCG. Thank you very much.
Sanjay, thank you. Those are all good questions, and really great to hear your support for this work. I'm glad you could join. Glyn, I'll leave you a chance to speak.
Hi, Sanjay. So when I was chief scientist at APHA, we did talk with with AJ vaccines about this issue of production. And as you know, Badger BCG is produced by AJ vaccines, and the cattle vaccine that is being validated at the moment in the field trials is the AJ vaccine. In terms of your questions, I think, yes, it would be, I think it's up to the competent authority of each country to license the vaccine, but I think data sharing on safety and efficacy is really helpful. And I think there may be also a role for OIE in that respect. And I know that we are editing the manual discussing the issues around cattle vaccination, but obviously we can't add cattle vaccination to the manual until there is a licensed vaccine. But I think there's a forum probably through OIE to look at how you might do data sharing to allow different regulators to license BCG. So I think there is a way forward there.
Thanks, Glyn. That's really helpful. Now, I was aware. Oh,
as well, I would think, Helen, that sharing of data.
Exactly.
And yeah.
Absolutely. Well, I think in a post-COVID world, there's a different perspective on these things, isn't there? On national, on globally sharing of data. No, absolutely. Colin, you've been waiting patiently.
Thanks, Helen. Thank you for the talk as well, Professor Hewinson and Professor Miller. Although Professor Miller did talk about, we've got to go and look at the disease first, but I'm sure you would possibly agree, both of you, that obviously we do need also that surveillance, because obviously, as we know, surveillance in some of the lower middle income countries is extremely hard to get. What's your thoughts regarding, yes, we've got to look at the disease, totally get it. We've got to look at the infection rate, totally get it. We've got to look at the population, how it's spreading and why it's spreading, totally get that. But we also need effective data sharing and effective surveillance of that. Your thoughts?
Great. Thanks, Colin. Glyn.
Yeah, I quite agree. You need to do surveillance on a number of levels. 1, to know where the problem is. 2, to know how big that problem is, and then 3, to see how effectively any intervention, whether it's vaccination or any other sort of control tools, are, how effective they are when you apply them. So I think without a surveillance system, it's quite difficult to implement any level of control. And I think that was one of the 10 pillars of the roadmap.
Great, thanks, Colin. I'm just going to pause you actually and move on, because we've got, if you don't mind, because we've got a couple of questions in the chat that I want Glyn to be able to get to. So question from Tom Ford: to what extent can host genetic diversity impact either vaccine, strain dependent efficacy, or DIVA diagnostic sensitivity? And if so, how could that information be interpreted for the benefit of industry?
Yeah, so yeah, thank you for the question. It's clear in terms of breed, it's quite difficult to know what the impact of breed is, and you actually have to do the experiments, I guess, and that's partly why you do field experiments with different breeds to see how both the test and the vaccines work. In terms of susceptibility, there does seem to be a greater susceptibility in Holstein Friesians to infection than Zebu cattle, about twice as resistant, but we don't know how that will play out in vaccination yet. But there are experiments ongoing in Ethiopia looking at crossbreeds and the different types of breeds, so that will help. Obviously, the worry would be if different breeds saw different antigens, and therefore your skin test efficacy is lowered. But that's really why you go into the field, to do those sorts of experiments.
Absolutely.
Share them. Yeah.
So there's another question here. So Halid is commenting, you, and I'm not sure if this was my comment or yours, Glyn, seem to conclude that the use of BCG vaccine in cattle in developing countries is currently impractical due to logistic and cost challenges. Can you elucidate why you think so?
Yeah, so I'm not sure it is, but I think we discussed things about cost, and obviously you'd want to have your production of the vaccine in, ideally in countries in which the need is there.
Absolutely.
But you also do need the infrastructure to be able to deliver those vaccines, and some sort of surveillance system as well. So all three of those really have to come together for some sort of effective control strategy, I think. Absolutely.
Well, it's the same as in human TB, actually. A vaccine is only part of the solution, actually. Yes.
Yeah, I can agree more.
So we have two more hands up now. So the first hand up was Graham.
Yeah, hi. Very interesting presentations there. I do have some concerns about some of the ways we're moving towards things like wildlife vaccination. For example, the East Sussex badger vaccine trial. We've got a very multifactorial disease affected by cattle genetics and cattle movements and badger densities and so on. We don't have many of the answers of how many badgers there are or where they are. And we're going to be vaccinating, and our readout is a fairly far removed proxy measurement of cattle bovine TB breakdowns. And it worries me slightly that we're looking at a very remote readout when we don't know many of the intervening steps. We're not going to be looking at, are we reducing badger TB levels, because we're not going to be taking bloods and testing and so on. And how do you think we could potentially overcome that problem?
So Greg, yeah, hi, sorry, Greg. I was distracted. Yeah, I don't know much of the details on the. I probably only know as much as you do from the press releases on the Sussex badger trial. I agree that if it is, my understanding is it's a pilot to look at logistics rather than a field trial to look at efficacy. But if you were looking at if you were to do a proper scientific experiment, obviously you would look at what the effect of the vaccination has been in the badgers in order to make any sort of conclusions about what had happened in cattle. But I'm not sure that that is the objective of that study.
Thanks, Glyn. Okay, so we now have a question from Mart.
Thank you, very interesting. So I really enjoyed hearing about the history of BCG. So, but I still think that we should give more emphasis to really the transmission. And so one of the ways that the second speaker was addressing this whole new number of species that can be infected and could be a reservoir for BCG. So when you would reduce transmission in cattle, then you could have a key role in reducing the problem, especially also the onward transmission to humans. So I think that I mean, it's too little known and too few people consider it, but I think we really should be looking more at the transmission after vaccination.
So I agree completely with Martin. I know that we've been on committees together looking at these things, but I guess the issues are that these are very difficult experiments to perform and are pretty expensive if you're going to do them in sort of UK sort of facilities. I think some of the exciting facilities that are now being set up in Ethiopia will allow us to get a better handle on the onward transmission work.
Yeah. Thanks, Mart, for that comment. Very good. Sanjay has come back just to ask quickly, Glyn, and then I think we might have to make this the last one. Does high incidence of TB in developing countries indicate a higher need to vaccinate cattle in these countries?
Difficult to know. You need to do the epidemiology, I would say. So, but I think some of the some of the whole genome sequencing pipelines that are now opening up are going to give us lots more insights into the sort of transmission pathways.
Yeah.
But I think what we are seeing is that high incidence of human TB, as Michele alluded to, is creating reverse zoonosis. So we are seeing humans giving animals TB.
Yes. Yes. No, that was very interesting, wasn't it?
And that's
all right.
Ethiopia, China, India. We're seeing it in many places now.
Really interesting. Two-way transmission. The last question, and I will make this the last one then, is from Jordan, who has talked about feline TB. So we see several cases of bovine and bovis infection in cats, and this is in the UK. And could vaccination of cattle and badgers reduce cases of M. bovis in cats, but would there potentially be the risk of BCG infection in those cats? Obviously, BCG is an attenuated organism, so less likely to cause infection than bovis.
Yeah, I mean, cats are very susceptible to TB, and often you'd use a farm cat as a sentinel to know whether you had TB on that farm, and we have seen some explosive, we have seen infections in cats either through wildlife sources or through them being fed milk in the past. In terms of there is an increase, if cats get M. bovis, obviously there is an increased zoonotic risk. I think the risk of BCG infection in cats is pretty low because the doses that they would be likely to be exposed to would be 0 through milk, because those studies have been done. So it's very difficult to conceive how the cats would be exposed to BCG.
Yeah. Super. Thank you.
Is negligible.
Treated. Yeah. Brilliant. I think we will draw it to a close there. It's now 7:30. Really, I'd just like to thank Glyn and Michele. I don't think we have Michele back, do we, Blakely? But we can email her afterwards. Really like to say thank you to you both for a really fascinating couple of talks, and clearly very interesting debate afterwards and discussion. Thank you very much.